Today I'm going to introduce DirectSeq Biosciences, an RNA sequencing biotech startup company. Many of you are familiar with the Human Genome Project, which took over 13 years and billions of dollars to draft the first sequence of our human genome. Similar in scale, today's Human RNome project will sequence all human RNAs with every modification. RNA, however, is more complicated than DNA because there are so many possible modifications. In addition to the four basic RNA nucleotides, A, U, G, and C, which mirror the DNA bases, we now know that there are more than 170 different types of RNA modifications which can completely change the function and stability of RNA. To complete the RNome, we will need a method that can sequence all these RNA modifications, a method which is not currently commercially available. Therefore, there is a growing call for scientists and organizations to coordinate a grand-scale Human RNome project to push forward academic research and pharmaceutical development into the next frontiers. A huge gap exists between what is needed for sequencing the Human RNome and what is available. The vast majority of current RNA sequencing methods were not designed for reading modified ribonucleotide bases, and those that can fail to capture all modifications simultaneously. Instead, separate methods must be applied to capture each modification type, one modification at a time. This applies to cDNA-based methods such as RNA-seq, which are indirect sequencing methods in that they require reverse transcription for converting RNA into DNA prior to sequencing. During this process, most modifications are lost or only inferred indirectly. Even with specialized workflows, collectively, about 16 modification types can be mapped, which is about 10% of all modification types discovered so far. Nanopore sequencing is more direct, but introduces a different limitation. The signal generated depends strongly on local sequence context. This means the same modification can produce different signals depending on neighboring bases. As a result, modification identification becomes ambiguous, especially in regions with clustered modifications. So despite major advances, existing technologies cannot comprehensively solve the RNome problem. DirectSeq Biosciences, in collaboration with Dr. Shenlong Zheng's lab at SUNY Albany, has developed a fundamentally new approach, NextGen MassSpec-Seq. This is an all-modification sequencing platform that allows sequencing all modifications simultaneously to uncover ground truth RNA sequence for RNome sequencing. For sequencing, a signature tag is needed to identify each nucleotide. 4 different colors are used in cDNA-based RNA sequencing, and electrical currents are used in nanopore-based sequencing. Instead of color or current, our method uses mass as the sequencing readout. Nothing is better than the mass, which is the natural tag both for canonical and modified nucleotides, including potentially novel or unknown modifications. These would be very difficult for current methods to identify. Sanger sequencing is a de novo sequencing method because it relies on a ladder generated by the Sanger dideoxy reaction. With NextGen MassSpec-Seq, we generate similar ladders for sequencing RNA using highly controlled formic acid hydrolyses, which cut RNA once at random positions along the RNA. This generates 2 pools, one pool for 5' ladders and the other for 3' ladders. These pools are then measured by LCMS. Base calling is made by comparing the mass differences between the 2 adjacent ladder fragments to unique unit masses, which are 306 Daltons for uridine, 329 Daltons for adenine, 345 Daltons for guanine, and 305 Daltons for cytosine. Proceeding pair-to-pair allows us to read each nucleobase in the RNA. Here is what our reads typically look like. We can see 2 clear sigmoidal curves of RNA ladder fragments in the mass versus retention time plot. Once we find ladders and their ladder fragments, we can then de novo and directionally sequence the RNA, reading the sequence from the 5' to the 3' direction or from the 3' to the 5' direction. The major advantage of this method is that we can not only read regular nucleotides, but also read essentially all the nucleotide modifications simultaneously. However, the early version of this method had a limitation in that it could not handle complicated real RNA samples. To address RNA mass data complexity, together with the Zeng Lab, we have developed multiple patented technologies using computational data separation for sequencing. In complex RNA mixtures, each RNA species produces a distinct trajectory in retention time, mass, and intensity. These trajectories form characteristic patterns that can be separated computationally, allowing reference-free sequencing of heterogeneous RNA populations. NextGen MassSpec-Seq is a truly direct and a truly de novo method for sequencing RNA. Our method does not require cDNA conversion, library preparation, or reference sequences, which could complicate RNA sequencing. Our method can now sequence different cellular RNA and therapeutic RNA ranging from 20 to over 2,000 nucleotides in length, including cellular microRNA, tRNA, small ribosomal RNA, and even therapeutic messenger RNA. Sequencing of other RNAs are also under development. Shown here is one tRNA we've sequenced recently. As you can see, we can read not only the canonical bases A, C, G, and U, we're also able to read the modified bases, including pseudouridine. Among 7 modification sites of this particular tRNA sample, 3 of them are not modified in every molecule. That is, they exhibit partial modification. Computational data separation allows us to not only identify each modification in its primary sequence context, but quantify isomers containing it stoichiometrically. And to our knowledge, this is the first demonstration of this kind of capability. The tRNA sequence shown in the last slide was from a specific purified tRNA, but our method can also handle complicated total tRNA samples. NextGen MassSpec-Seq is capable of uncovering ground truth tRNA sequences with all their modifications at single isodecoder resolution, which is currently very challenging for other methods to accomplish. When applied to therapeutic small guide RNAs, we achieve full-length end-to-end sequencing. Importantly, because this method does not rely on a reference sequence, we're not limited to sequencing of the intended target sequence only. In fact, we discovered significant quantities of impurity RNA sequences coexisting in the small guide RNA sample. NextGen MassSpec-Seq is thus capable of elevating small RNA drug quality control in a manner that is more accurate, convenient, and comprehensive than traditional approaches. We also extended our method to sequence larger RNA molecules, including most recently a 1,937 nucleotide therapeutic mRNA provided by NIST, demonstrating that for the first time we can use mass spectrometry to directly de novo sequence full-length therapeutic mRNA. Shown here is our sequencing result of the mRNA's poly-A tail sequence, which cannot be directly sequenced by other methods. When sequencing this NIST reference mRNA, we identified unexpected sequence variations, including significant amounts of different cap structures, resulting from inefficient conversion during the multi-step cap1 installation, including addition of the M7G PPP cap and 2' O methylation of the first nucleotide. This provides insight into features that are critical for RNA stability and translation. While there are so many things we can do with our direct RNA sequencing, what is most inspiring is the ability to provide ground truth RNA sequence with respect to every modified base. With this unique capacity, our initial aim is to help sequence the first human RNome and provide the highest quality control standard for RNA therapeutics. This is the path we propose for sequencing the first human RNome. With our ability to determine the true sequence of tRNAs, we now have the solid foundation for tRNome sequencing, which is about 10 to 20% of the human RNome. Now underway to extend this approach to other RNA types like ribosomal RNAs and even messenger RNAs, we move closer to completing the entire RNome. A mass spectrometry method like ours is likely to represent a core technology for achieving the first human RNome sequence draft, much like Sanger sequencing was for the Human Genome Project. High-throughput methods like NGS and nanopore sequencing will also play a crucial role, providing a canonical, or ACGU, primary sequence core of this draft. By directly sequencing heterogeneous RNAs without prior sequence input, our method addresses key limitations of current RNA analyses, providing a powerful tool for RNA drug development, quality control, and regulatory validation. Our model is to begin as a centralized sequencing service, then expand to on-site deployments with partners. and ultimately deliver a fully integrated platform, including instrumentation, reagents, and software. The value proposition of our integrated platform is straightforward. Academics will be enabled with better tools for understanding cellular and genetic control. Essentially, they'll be able to largely fill the epitranscriptomics gap in our current transcriptomics understanding. Drug developers will be able to leverage the platform to develop more powerful drugs, enhance their quality control procedures, and reduce clinical trial risks. Patients will experience better outcomes and enhanced safety. For regulators, they'll find that the refined critical quality attributes simplify their job. And for national security and defense, we'll be able to better attribute exotic viral RNA sequences in the field. This technology has been published in multiple peer-reviewed journals, including Nature Communications, JACS, Nucleic Acids Research, and ACS ChemBio. Support from NHGRI through SBIR Phase 1 awards has been critical in advancing this platform from concept to small RNA sequencing, all the way to most recently full-length mRNA and cellular RNA analysis. We welcome you to join our efforts in growing DirectSeq.